Retinotopy describes the neuronal wiring between the eye and the brain in all animals with image-forming eyes. In adult Drosophila, this mapping is somewhat special because the fly eye consists of about 800 individual single eyes called ommatidia, and there are 6 photoreceptors in each of those ommatidia that contribute to the image formation in the first optic neuropil, the lamina, shown here in the brain. But these 6 from the same ommatidium do not actually, in the adult structure, end up in the same place. This so-called neural superposition visuotopic wiring develops from an input pattern of ommatidial axon bundles that get resorted into the visuotopic wiring pattern, and that output pattern then maps neighboring points in space as neighboring points in the brain. Developmentally, however, if we now look at the developmental process in reverse and remove the output pattern and look at the temporary sorting plane where the initial axon bundle was resorted and go back to the initial axons coming from a single ommatidium, we're looking at the actual temporary retinotopy. So shown here is just one of those pixels that is mapped with 6 photoreceptor axons temporarily to one point in the brain. And these 6 photoreceptor axons are arranged 800 times in a mirror-symmetric fashion. The so-called equator line separates the dorsal and ventral halves of the eye, and this equator line is randomly zigzagging in each individual eye. And the starting point for our study was to show that in the temporary brain structure, this random zigzagging line is perfectly preserved. So we asked, how can this precise pattern in the developing retina be preserved in the brain? And so for this we need to look at something that has been studied in developmental neurobiology for many decades, the development of the so-called eye disc in Drosophila, where a wave of differentiation, shown here as this red bar, the so-called morphogenetic furrow, sweeps across an epithelial tissue of undifferentiated cells and in its wake causes the differentiation of photoreceptor cells in a certain order. Our study now contains evidence that suggests that the axon outgrowth underneath this epithelium follows the same wave of differentiation. So on both sides of the zigzagging equator line, the first photoreceptors that grow to the brain are, after the so-called pioneer axon, photoreceptor 8, the green axon shown here, 2 and 5, followed by 3, 4, and 1 and 6. Row by row, each of those outgrowing axons wrap around a dense optic stalk and thereby preserve the organization from the eye before they defasciculate in the developing lamina plexus. Here, the axons regenerate the pattern that has been originally defined by differentiation in time in the eye, and key forces help organize this pattern. Shown here is the inter-bundle adhesive force mediated by the protocateran flamingo, whereas the intra-bundle adhesive force that keeps 1, 2, 3, 4, 5, and 6 from an individual bundle together is the homophilic cell adhesion molecule sidekick. Together, these inter-bundle and intra-bundle adhesive forces can preserve perfectly the pattern that is originally generated by a wave of differentiation in time, row by row, in the developing retina. And that includes the randomly zigzagging equator in the eye that can be found in the precisely same pattern in the brain. To show that these two forces are sufficient to recreate the retinotopic pattern in the brain, we turn to a soft body modeling shown here using Unity Game Engine. Think Angry Birds of visual map formation. This simulation recapitulates the arrival of axon terminals, first of R2-5 in green, followed by 3-4 in yellow and red, and 1-6 in blue. And the only forces included in this simulation are the inter-bundle flamingo and the intra-bundle sidekick adhesion, with row by row arrival of axon terminals in time. The gaps you can see between the photoreceptor axon terminals were not encoded in the process, and if we label the postsynaptic laminar neurons, shown here on the right in white, and compare them to the shape of the gaps left by the patterning process, we find they're precisely matched. We conclude that a temporal gradient of photoreceptor differentiation and row-by-row axonal outgrowth, together with two adhesive forces, can ensure retinotopic map formation, preserving the precise pattern from the eye into the brain, including the zigzagging equator line shown here, without a target-derived organizing principle or mechanism.